Buffers are a small portion of this unit. It's very, very small. That you just kind of have to understand what they are. We don't do any calculations or anything with them. But a solution which resists changes in pH is called a buffer solution. In English, the word buffer means to resist change or protect or lessen the shock. So in chemistry, the buffers will resist a change in pH. Okay, you have buffers in your body. Your pH of your blood runs about 7.35 to 7.45, and in biology you learned that H word for maintaining a buffer solution or a buffer. Remember what it was? Homeostasis. Okay, but homeostasis is your body has ability to keep its blood at a certain pH level. Otherwise, it would be detrimental. If you get too acidic or too basic, you can become very ill or even coma or death. Okay, so it's not good. But for a buffer to work, it must be able to resist acid and bases. So it resists the changes that they would cause in the pH. They're made by adding a weak acid or base to the salt of its conjugate. So it's going to be weak acids and weak bases that form buffers, not strong acids and strong bases. Okay, because we'll learn in a second that a strong acid and a strong base is a complete neutralization. So basically, for a test, you just kind of need to know that a buffer resists a change in pH, and it's formed by a weak acid or a weak base. Okay, and it's salt. That's the important part. Okay, so here's kind of what that system would look like. We're just going to let you glance at that. It's not something we're going to spend a lot of time on, but you know that strong acids and strong bases, remember, they fully dissociate, and so they fully break apart into their ions, whereas weak acids and weak bases do not. And so because of that, there's a different kind of reaction going on. And so when you have a weak acid and it's salt, it's something called the common ion effect, and it can resist those changes in pH. And that's all we're going to say about that. Water is special. We talked about this when we were doing conjugates. Remember that water is something called amphoteric. Second time I've given you that definition, so it kind of might be important, right? I've given it to you twice now. Sometimes it's called amphiprotic. They mean the same thing. Someone just decided to give it a different name. But remember, that meant they could act as an acid or a base. And water itself is neutral, okay, because it has this ability to auto-ionize or self-ionize. And looks kind of like this. I have a water molecule plus a water molecule, and it would give me H3O plus OH minus. So one of those water molecules would act as a base, the other one would act as a acid, and then on the other side you'd get the conjugate acid and the conjugate base. Okay. On one of those worksheets we had that formula at the top, and I told you to ignore it, where it said Kw was equal to the hydronium times the hydroxide is equal to 1 times 10 to the negative 14. That's where we get, remember our pH scale was 0 to 14. That's where we get the 14, because together those multiplied by each other are, if you take the negative log of that value, gives you 14. So when you have an aqueous solution, you actually have hydrogen and hydroxide ions present. If you have an acid, you just have more hydrogen ions present and less hydroxide. If you have a base, the scale switches and you have more hydroxide and less hydrogen, but you still have both, okay? And that's because no matter what, and it is a constant, no matter what your concentration is, if you multiply them by each other, it's going to equal that 1 times 10 to the negative 14, okay? So when you have a neutral solution such as water, your hydrogen is equal to 1 times 10 to the negative 7, but guess what hydroxide is also equal to? 1 times 10 to the negative 7. And if you were to take the negative log of either one of those, you would have 7 pH or 7 POH. Okay? So you do need to know that water is amphoteric. You need to know that it can auto-ionize. So you need to know this, and you need to know that it's neutral, and you probably should know that constant. So those are the things you need to know for a test. Okay? So indicators are going to be the next thing we talk about. And indicators are a special kind of chemical that change color in the presence of an acid or a base. So in other words, if it's an acid, it might be one color. if it's a base, it'll turn into a completely different color. Okay. We're going to do a titration tomorrow in the lab, and we're going to be using an indicator of bromethymol blue, okay, which is going to be blue in the presence of a base. And in the presence of an acid, it's actually yellow. But right here in the middle is the sweet spot, which we're going to be looking for is neutral, is green. A lot of you will not get green because it's very easy to overshoot a titration. Got to go really slow at the end to get green, okay? But that's what we're going to be using. You've heard of litmus before. In our characteristics, we said acid, it's red litmus, and base is blue litmus. Okay, so you need to know that for a test. Has anybody ever seen pH paper that like changes colors as it goes up the scale? That's basically paper that's been soaked in a universal indicator, so it changes colors at each level along the way. It's not as accurate, but it is useful to know a range, a rough range. Phenolphthalein is a very popular one. It's actually colorless in the presence of an acid, and it turns a really pretty pink in the presence of a base. And then you even have things like fruits and vegetables, flower petals, dyes, and color film. There's all kinds of indicators out there. Okay, there's one red cabbage that you can use as an indicator. In nature, there's also things that change colors based on their soil, pH, and things like that. These are just some common ones. The color scheme somehow on the projector is not quite the same as it is on my screen because the pink looks red and it's actually pink for phenolphthalein. But here is the neutral point. So you pick an indicator based on where your pH range is going to change. So tomorrow we're doing strong acid and strong base. And so it's going to be right here in the 7 range. So that's why we're going to use bromethymol blue because that's where its color change happens. Okay. If you were doing something that changes colors, say if you're doing a strong acid and a weak base, you would use a different indicator. So you pick your indicator based on where you think your pH is going to be when the change happens. Okay. This is just to show you how you have acid-base indicators in nature. Anybody have hydrangeas in their yard, or you know what flowers I'm talking about? Okay, It's a bush, but it has a flower on it. Okay, the color of the flower depends on the pH of the soil. If the pH of the soil is acidic, you'll get the pink flowers. If the pH of the soil is basic, you'll get the blue flowers. And you can actually change the colors of the flowers based on messing with the pH of the soil. You can add different fertilizers or additives to your soil to get it whichever one you would like. The blue seems to be more common, especially down here in the south. We looked it up because my pre-AP class was curious. If you have a neutral pH, your flower would be purple, right smack in the middle, which I would think it's really hard to maintain a neutral pH of your soil, but okay. Neutralization reactions, that's what we're going to be doing when we react a strong acid and a strong base. This word goes right here. Write that word in there. It's the reaction of an acid with a base. Remember, it produces water and a salt. Okay. Notice we're not saying NaCl there. Please know that when I say salt, I mean it's a whole class of compounds, not just NaCl, table salt. Because invariably a question might say, identify the salt, and they'll come up there and they'll show me the paper and they go, but there's no NaCl on here. And I can't on test tell you. It's because that's not just the only salt there is. There's a whole class of compounds, okay? And it's any time an acid and a base react together, okay? When they do, we get the salt, and what it is it is the positive cation that comes from the base, and the anion, the negative, comes from the acid, okay? And then, of course, the hydrogen comes from the acid, and the hydroxide comes from the base to form that, okay? When a strong acid reacts with a strong base, they produce a neutral salt, so they'll be neutral. And that is the only kind of neutralization reactions we'll do in this class, is strong acids and strong bases. They're the easiest. If you start getting into weak acids and weak bases, there's a whole other type of calculation that you have to do to get the pH and see what's really going on. And we do that in AP Chem, but we do not do it in here. Okay? But that's the general formula. And then I have a couple of examples. just to kind of show you that, yeah, this is the classic example that we use, and it's forming NaCl, but this is also an example of an acid-base reaction right here, and this would be the salt that you would get. Okay, I could do HBr plus KOH. Both are strong acid and strong base, and I would get KBr plus water, and then that would be my salt. So when you see this reaction, pretty much whatever's not the water on the product side is your salt. So don't let that confuse you on a test that there's no NaCl. Okay? So that brings us to titrations. This is the last little section of the notes, but this is the lab that we're doing tomorrow. So please wear the appropriate shoes tomorrow, because we are going to be working with acids and bases, so no flip-flops. I got to remember to wear the right shoes. Sometimes I forget. Okay? So titrations is a technique that is utilizing that neutralization reaction, because remember I said it forms water and salt. Okay? And so if we know the concentration of one of our items, we can figure out the concentration of another by knowing the exact volumes of each that we're using. Okay, the known concentration we call the titrant, and it is usually located in the burette, which I'll show you a picture of in a second. If you've peeked into the lab in the last day or two, you've seen those long tubes on the little holders. Those are burettes. They're a very, very accurate and precise volume delivery system. They're to 0.01 milliliters. Okay. And so what gets confusing is how to read them, so we'll talk about that here in a second. The end point is the point at which that indicator changed colors. So for us, we're looking for green tomorrow. Most of you will probably get yellow. Titration is a very, it's an easy technique, but it's got to be very precise at the end point. And usually kids get a little too hurried to try to finish it, and they overshoot the end point and they go to yellow. Okay? And when you get to yellow, it's going to basically look like the color of urine. It's that color yellow. Okay, maybe a little bit darker than your urine should be, but it is going to be yellow. If it's green, it's going to be a real pale green. If you get green, and it's sudden. One drop can make the difference between blue and green and yellow. It's very endpoints are very sensitive, because it stays blue as long as there's one molecule of base. Okay, as soon as you have one extra molecule of acid, that's when you start going over and bam, it's yellow like that. It's that sensitive. Okay. The equivalence point is the point at which they are actually 100%. I have the same amount of hydrogen as I have hydroxide. Okay. The only way you can truly see an equivalence point is if you're running a pH with the pH meter in there that's measuring the pH the whole time. Okay. The end point is going to be slightly past the equivalence point. And when I mean slightly, I mean half a mil, 0.1 mil, just slightly past, just enough to make it change colors. So it's an easier process, it's much faster, and it doesn't make a huge difference in our calculation. Okay, but please know that the endpoint is slightly different than the equivalence point. Okay, the endpoint's just where the color change happened. These are the steps involved in the titration. I'm going to want you to read those before tomorrow. That's your only homework tonight, is read the titration stuff so that you kind of know what's going on, because it is a different technique. When you come in class tomorrow, I'm going to show you a demonstration of what it looks like, so you can kind of see what that should look like. And hopefully I get green, because sometimes I do it too fast up here and I overshoot to yellow. So hopefully I can show you what that looks like. And then I'll overshoot it to show you what it looks like if you've overshot your titration. This is what your setup looks like. Okay? Only we're not using pink, and we'll have blue. The burette, when you look at it, it's going to be different than what you're looking at when you're used to reading volumes. The top of the burette starts at 0, and the bottom of the burette starts with 50. And so when it's full, the meniscus is going to be on 0, okay? Because it's measuring how much volume you deliver, okay? How much volume you deliver. And so let's say I walked up to my burette, and it's like right here. It's actually at 1.1 milliliters. So I would write that down as my initial. And let's say I did my titration and I used to about right here. And I said, okay, it ended at 15.7 milliliters, and that was my final. So I would subtract the 2, and I would say that I delivered 14.6 milliliters used. So that's how you read those. Okay? To calculate, because remember I told you could calculate the unknown, you're going to use this formula. MAVANA equals MBVBNB. The MA and VA is molarity and volume, just like they have been in the past. So this is the molarity of the acid, the volume of the acid. Molarity of the base, volume of the base, just like what we know what those letters stand for. The Na, however, is moles of hydrogen in your acid. The Nb is moles of hydroxide in your base. So we're not going to be looking at the balanced equation. We're just going to be looking at the formula of the acid and the base. Okay? So for instance, if I have HCl, my Na would be 1 mole of hydrogen, because there's 1 hydrogen in my formula. If I had H2SO4, my Nb is going to be, not Nb, Na, sorry, 2 moles of hydrogen, and that's because of this 2 right here. Okay? Oh, I needed to, all right.